So now we'll move to the active speaker portion of our program. The first trio of speakers are SWOG executive officers who oversee our disease committees which run treatment trials. I've asked these EOs to select the most innovative trials going on now in the committees they oversee and to report on those studies today. Our first EO is Dr. Julie Gralow, who again oversees the breast and lung committees. Julie, welcome back to the podium.
Thanks, Chuck. I've chosen one primary study within breast and one primary study within lung to overview. These are studies that keep giving and I think have many innovative qualities to them. Within breast cancer, I chose the SWOG 8814 study, which is an old study that keeps on giving. It was an adjuvant chemohormonal therapy study for estrogen receptor positive, lymph node positive, primary breast cancer. Kathy Albain was the principal investigator and provided me with most of the slides I'm using here, and she's listed her following colleagues who have been part of a lot of the subsequent work that's gone on with this trial. The original backbone of the trial, and it has an 88 number, so it got most of the way opened by 1988, were postmenopausal, lymph node positive, estrogen receptor positive breast cancer patients, stratified by nodal status, either 1 to 3 positive or 4 or more, progesterone receptor status, and the time from surgery. And then patients were randomized to receive the standard of care, which was tamoxifen at the time, versus tamoxifen plus chemotherapy. And the chemotherapy, CAF, cyclophosphamide, doxorubicin, and 5-fluorouracil, was either given concurrently with tamoxifen or was given first, and then tamoxifen was started after the chemotherapy had finished. About 1,500 patients enrolled, and the primary outcomes are shown here. The first objective was to see if CAF chemotherapy added to tamoxifen in ER-positive, postmenopausal, node-positive women, and the answer was, when we combined the two chemo and tamoxifen arms and compared it to tamoxifen, that yes, chemo added in this broad population. Chemotherapy plus endocrine therapy became the standard of care for ER-positive, node-positive, postmenopausal patients, in part based on this study. The second objective was looking at how the chemo and the tamoxifen was given, again looking at concurrent CAF with tamoxifen versus finishing the CAF chemo and then starting the tamoxifen. And sequential chemo followed by endocrine therapy was found to be favored and became the standard of care. So whereas prior to that, some people started both together, the chemo and the endocrine, others waited till the chemo was done. Now, this study showed us that we should wait and sequence, and that outcomes were actually better if we didn't give chemo concurrently with tamoxifen. This shows curves from the 10-year disease-free survival, 10-year overall survival, where the yellow, which is the curves that are doing the best for disease-free and overall survival, would be the sequential CAF tamoxifen arm. The red color here is the concurrent chemotamoxifen, and the blue is tamoxifen alone. Now, of course, there are more toxicities that came along with adding the chemotherapy, wouldn't surprise any of us, and we documented the increased neutropenia, stomatitis, thromboembolic events, congestive heart failure, and leukemias that occurred. And in part, this was one of several studies that led to this conclusion from an NCI Consensus Development Conference on Adjuvant Breast Cancer that was published in the year 2000. And studies like this that showed that in general, chemo added to tamoxifen resulted in a statement from this consensus conference that said chemo should be offered to the majority of women with early stage breast cancer, regardless of the tumor size, the node status, their menopausal status, or their hormone receptor status, because we don't know how to pick out who doesn't benefit. So that was the conclusion. And of course, as soon as we made that conclusion, we knew we were wrong. Not everybody really needed it, but we knew it was right that we didn't know how to pick out who didn't benefit. So we've moved into the present and the future, where we're now individualizing estimates of recurrence risk and estimates of chemotherapy benefit using genomic profiling. And indeed, we have been able to sort out that not all patients and tumors benefit from chemotherapy. This is work done by our colleagues at NSABP, now NRG, that showed in a node-negative population that we could look at a 21-gene recurrence score assay using RT-PCR, and we could pick out a low-risk group of ER-positive, node-negative patients who did fine with tamoxifen alone, and we could pick out a high-risk group from that same population where chemo absolutely benefited the population. So this was a first step, and it led to the design of a prospective trial, because that was all based on retrospective data, called the TAILOR-X trial that we participated in. Dan Hayes was our SWOG co-PI, and this was a prospective trial looking at using the recurrence score to randomize patients to get endocrine therapy alone, chemotherapy plus endocrine therapy, or to randomize in the middle to hormone therapy plus or minus chemo. We've now seen reporting of this very low-risk group in the node-negative setting that shows that at almost 70 months of follow-up, that distant disease-free survival in this group that got endocrine therapy alone is 99.3%. Can't really do better than that. And my slide is stuck. If we could advance it to the next slide. Thanks. So, knowing that data in the node-negative population, Kathy and the SWOG breast group said, we have a study where we have node-positive patients, and we could ask a similar question as to whether we can use biology, whether we can use genomic profiling to actually predict for recurrence and benefit of chemo in a node-positive population. So that SWOG 8814 study was used, and I'm stuck again. And because there had been an optional tissue banking protocol as part of the study, we had tumor on almost 50% of the patients. This was optional. It was done in an era where we didn't know what we were going to do with it, but we collected the tumor, and then many years later came up with an idea for what we were going to do. So we were able to collect the tissue and perform the RT-PCR 21-gene assay in almost half of the patients, and this shows the numbers for that we only had about 9% of tumor blocks that were quite old at this time that we couldn't do the RT-PCR assay on.
We have a backup clicker.
Great. Thanks, Wendy. And this shows data from that RT-PCR genomic profiling analysis of the SWOG 8814 patients. The first thing that we were able to show was that the recurrence score assay provides prognostic information in patients who were on the tamoxifen alone arm. And so you can see in red a low-risk group by the recurrence score assay, blue the intermediate risk group, and yellow the high-risk group. Disease-free survival and overall survival separates out. And then, equally important to having prognostic value, we showed that there was predictive value in this ER-positive, lymph node-positive group, where in the low-risk group with a recurrence score at the low end, here in this case less than 18, you didn't see any statistical difference for giving chemo or not. But in the high-risk group with a recurrence score greater than 31, we did see a statistically significant improvement in disease-free survival by adding chemotherapy. So even in a node-positive population, we were able to sort out a lower-risk group and a higher-risk group and predict which would benefit from chemotherapy. Now, the translational work from SWOG 8814 that I've just showed you led us to the SWOG 1007, or the responder trial. That's an important trial that's essentially met accrual in the US. We're adding a few more French patients on. But this is a prospective assay looking at patients with a low to intermediate recurrence score and randomizing to endocrine therapy alone or endocrine plus chemo in the node-positive population. So this study has essentially met accrual in the US, but it's going to be years before we get the results of this. Translational work from SWOG 8814 also led in part to the current ongoing SWOG S1207 trial, which is a trial looking at the patients who are ER positive and have high risk, and we need to do better. So in part, by using the recurrence score in this high-risk group, we are screening for patients and enrolling patients who would have a higher risk of recurrence from endocrine therapy alone or chemo plus endocrine therapy, and we're looking at adding everolimus, an mTOR inhibitor, for a year or not to standard endocrine therapy to see if we can do better in this group that's not doing so well. So we can avoid adding the targeted therapy to the group that is doing well. And most recently, we've seen from SWOG 8814 that we might be able to predict local regional recurrence, and what this slide shows is for a group of patients who have a mastectomy, have 1 to 3 positive nodes, what you see is if they have a low recurrence score and they don't get radiation, they do well, but if they have a high recurrence score and they don't get radiation, they do poorly. So maybe we can use, and we'll need to evaluate this prospectively, the recurrence score to decide in that 1 to 3 positive node group who we add radiation to and who we can safely omit radiation in. So data from the SWOG 8814 trial led by Kathy Albain spans many decades and continues to provide information about adjuvant therapy decision-making in ER-positive, node-positive breast cancer. It's allowing the withholding of toxic therapies where little benefit exists, and it's spawned multiple subsequent trials. So the key lesson learned from SWOG 8814 is the benefit of prospective tissue collection for studies that could not have even been imagined at the time of the study startup. And Dan, I know I'm speaking your language. When we start up a study, he's saying, get all the data we can, get all the samples we can, because we never know 10 years from now what we're going to want to study. And this study proves it. Now moving on to the lung committee, the trial that is really many trials within a trial that I've chosen to highlight is the Lung MAP trial, or S1400, the Lung Master Protocol, with leadership with Fally, Roy, David Gandera, Fred Hirsch, Phil Mack, et cetera, listed on this slide. This is the lung map design. I think a lot of you are familiar with this. It's for squamous cell carcinoma of the lung, previously treated now and have progressed or relapsed through a first line of treatment. A centralized biomarker testing is sent off with genomic analysis, and depending on what that genomic analysis shows, patients are placed in different arms, depending on what's open at the time and what's available, or if they don't match any of the open marker studies, there's a non-match trial for them to enroll on as well. So there's really something for everybody. This is really a unique trial design, and I'm very impressed with the study and the leadership in it. So the unique features of the trial are the collaboration. It brings together multiple key stakeholders, industry, academia, patient advocates, and government agencies to advance precision medicine and development. This trial is addressing the 4 primary goals from the Institute of Medicine 2010 report for cancer clinical trials modernization. And those goals are to improve the speed and efficiency of trial development and activation, incorporate innovative science and trial design, improve the prioritization, support, and completion of trials, and incentivize participation of patients and physicians. Other unique features, this broad next-generation sequencing improves enrollment efficiency. Instead of testing one by one a variety of single markers by spanning and doing a broad testing up front, it improves the likelihood of receiving a drug targeted to the tumor's genetic profile and enhances the ability to study rare subsets of lung squamous cell carcinoma. The master protocol design allows new therapies to be added as the trial progresses and some therapies to be dropped out. It improves operational efficiency. The Singer master protocol is amended instead of having to submit new trials each time. And there's a unique lung map drug selection committee. Another unique feature is it's designed to facilitate FDA approval of new drugs, as well as the biomarkers that go along with them. And industry and the FDA have been at the table together from the beginning. If the drug meets its predetermined efficacy and safety criteria, the drug and its accompanying biomarker are eligible for FDA approval. So we have potential to get promising drugs to the patients faster. And the non-match arm allows all patients access to some form of interesting investigational therapy. Additional unique features include that this trial has allowed patients, even though the eligibility is you have to have progressed or relapsed after at least one line of therapy, it allows tissue pre-screening. So while you're on your first-line therapy, and even still responding to that, you can send the tissue in so that at the time of progression, we don't have to wait for the sequencing and we can immediately get the patient into an open arm. There's a very active site coordinators committee. I've been very impressed with this committee. It represents study site staff, nursing, CRAs, data management, regulatory levels, and they provide input into the study and help with accrual and help facilitate the study. And now there's a new important patient-reported outcome study being added to the immunotherapy arms. This shows the current status of the various sub-studies. It's a busy slide, but the first several arms have been closed down. New arms are pending or open. Currently, there's a PARP inhibitor arm open. There's a combined immunotherapy, non-match arm open, and several others in process. The biomarker results show that about a third of the patients are being pre-screened prior to their progressive disease, and about 2/3 are being screened at progressive disease. We see anywhere from 8% up to 19% positivity for a variety of biomarkers that have been tested in this study, and we see rare positivity for EGFR and ALK, which are important in our non-squamous lung cancers. This is data that was shown at the lung working group yesterday, and it shows the results from the first several arms of the trial that have been closed down. And unfortunately, the first several targets and drugs that were being tested had negative results. They did not do better than standard of care chemotherapy, but these are important negative findings. Even though there was an FGFR mutation, patients did not do better with an FGFR targeted drug compared to standard chemo. And we do have some responders in each of these arms, and there will be evaluation of the exceptional responders to try to sort out what allowed them to respond when the majority of patients in that arm, even though they had the mutation or the alteration, didn't. So one of the most important things, and we discussed this morning at an organizational meeting for the Lung MAP trial, are the lessons that are being learned from this trial. One, this unique private-public partnership is working. We have the NCI, the Foundation for the NIH, SWOG, and Friends of Cancer Research as the core key for primary partners. We've got all the NCTN sites that are active partners. We have 7 precision medicine and or biotech companies involved and multiple advocacy partners. The master protocol is feasible. The infrastructure facilitates opening new arms quickly, and the phase 2-3 design that it's evolved into allows rapid drug biomarker testing for detection of large effects. The promising drugs move on to phase 3, and we close down the non-promising drugs sooner. Sites and patients are interested in this study. We're screening large number of patients for multiple targets. 740 sites have IRB approval. 365 sites have at least one patient accrued, and we have over 1,200 patients that are registered, with 456 registered within a sub-study. Biopsies and broad screening with rapid turnaround by next-generation sequencing is feasible. Rapid tissue assessment and return of the NGS results is possible. We have shown a median of 1 day for receipt of the tissue and 9 days for the results reported back to SWOG, and greater than 90% of the samples are analyzable. The trial is able to be flexible and current. It's keeping up with the evolving treatment landscape. 3 studies have already been closed, 3 sub-arms. The questions are answered. Multiple new arms are open. It's changed from a randomized phase 2 format to the new phase 2 moving on to phase 3 format. And when 2 immunotherapy drugs were FDA approved in 2015 for squamous cell lung cancer, the trial rapidly was able to incorporate that, and immunotherapy became an important component of S1400. Research has been added to predict response and benefit from immunotherapies and to look at combinations of chemo and other targeted therapies and immunotherapies. And this is what Roy Herbst told me this morning was his most important contribution to the study, which is this funnel diagram that's showing everything that's going into the study with important information coming out. Additional lessons learned, adequate funding is needed. This trial gives extra funding to sites. They receive almost $6,000 for the screening and then registration of the patient. There are additional reimbursements for CT-guided and bronchoscopy-guided biopsies, and additional reimbursements for research-based procedures and on-site visits that are outside of regular appointments. It's expensive to do work like this, but this is what it costs. You can't do this without this kind of funding. And the future of S1400 is that the infrastructure of the study is allowing now the proposed expansion of this trial beyond squamous cell lung cancer, at least in the immunotherapy arm, where there are a lot of unmet needs and questions. So soon it's proposed that at least the immunotherapy part will expand beyond squamous cell lung cancer. And there's going to be expansion in leadership structure in the near future, incorporating more involvement from non-SWOG NCTN groups. So I would say I picked this study because it has so many lessons learned that we can hopefully apply to other studies. And I would say the S1400 study is a new paradigm for drug development and scientific discovery that hinges on collaboration at its core. Thank you very much.